Okay.
Okay, so the final talk of this session is by Joseph Dahdah, and the title is Optical Studies of Sub-monolayer dye films for photo alignment of liquid crystal cells.
So this work is done about 20 miles south of here in conjunction with the LC University here in Boulder. And I'll be talking about the same molecule in the last talk, this DMR molecule, which was synthesized here in Boulder by the Wall group. So generally I have to introduce this topic, but I guess the basic umbrella under which all this falls is why would you want to study dye functionalized surfaces? And I guess as nanostructures become more prevalent, ways of controlling surface properties become more important. And, you know, light controllable systems allow you to, have that kind of control over the surface of a nanostructured environment. The trade-off is that you have to worry about a lot of things. They're very complex systems. You have light matter interactions, intermolecular interactions on the surface of these dye structures, and you have to have really good techniques for even understanding what's going on, because like in my case, this is a monolayer, so it's a very small system to be studied. So I guess I'll just introduce the molecule a little bit. Azobenzene was pretty well introduced in the last talk. You have this trans-cis geometrical isomerization that you can control with light in the absorption band of the dye molecule. That pumping occurs in both directions. You can attach things to this molecule, and it doesn't really change that basic fact too much. This is good because then you can attach it to a glass slide like we've done here. And so this was developed by the Wall group, and I just do the last step of this chemistry to get a monolayer surface of these dye molecules for study. So in contrast to the last talk, I won't be investigating how the monolayer aligns liquid crystals. I'll just be studying the monolayer by itself. But it does align liquid crystals, and that's why it's of interest to this center. And this graphic here just displays sort of what happens if you have that twisted pneumatic liquid crystal cell that was introduced in the last talk. If you have a photo alignment layer on the bottom, and you have a sort of traditional nylon layer on the top, and they're both planar layers, planar alignment layers, then you can introduce polarized light, causing reorganization of the photoactive layer in whatever direction you choose, and thereby twist the liquid crystal cell. If you rotate that cell, then with respect to the polarization, you can change the transmission through the liquid crystal cell. I've got a little video of that. A lot of you have seen this demonstration in real life. This is the sort of schematic interpretation of it, but nobody's seen the movie, so I've got a little movie. And this cell was made by Ann Lowe, a new student in our group down in Mines. And there's a lot going on here, and there's blue spots in this cell. But basically, you can see the contrast that's developed when you rotate the cell 90 degrees. And then it goes dark in the condition where you don't have any twist in the cell. Rotate again, it goes light, and you have twist. So that's why this group is interested, why the center is interested in this photo alignment layer. Now, to study this photo alignment layer by itself without the aid of the liquid crystal to magnify the effect of photo alignment that occurs on the layer, on the monolayer, I thought that the best way to do that would be to study the solution first, because the solution is sort of a simpler system. You don't have to worry about orientation. You can just study the sort of photochemical properties of the molecule itself. And this figure is adapted from an earlier thesis on DMR, and basically shows the electronic absorption spectra of the DMR molecule in solution, and how the composite spectra that you measure, so this would be the spectra of a DMR solution that is being illuminated by the correct wavelengths of light. And that composite spectra is composed of two different electronic absorptions, one for the trans and one for the cis isomer. So the goal of these solution studies was to sort of characterize the photochemical quantities that would give rise to this kind of spectrum. And so these are things like cross-sections for the cis and trans absorption of the molecule, quantum yields for production of cis and trans ground states from excited states of the molecule, the thermal decay constant. And so I performed these experiments down at Mines, and these are the kinds of results you get. So basically the experimental schematic is that you're pumping the solution with a particular wavelength of light in the absorption spectrum of the trans molecule, that black curve. And that produces a certain fraction of cis in solution. And so you end up with a blue curve, like that one. And that corresponds to a certain amount of cis and a certain amount of trans in solution. If you do this for different wavelengths in a careful manner, as described by Rao, 1990 paper, you can actually extract the cis spectrum, the pure cis spectrum of the molecule, and that's what we did. And so that's this red dash curve. So now we know the pure cis spectrum of the molecule and the trans spectrum of the DMR molecule. And I also determined this quantum yield ratio, which basically says that for illumination at the trans peak absorbance, you're going to much prefer the creation of cis molecules than trans molecules. Remember, the light excites both trans and cis conformational change, in both directions. Other results of the solution study on this molecule indicate that the thermal decay constant for the molecule depends greatly on what solution you have the molecule in. So if you have a more polar solution, you get a different answer for the thermal decay constant. And there's actually a sharp transition somewhere as you get towards more polar solutions. So if you notice these less polar solutions in the top of this table, you get maybe a few minutes for the thermal decay constant, and then all of a sudden you get a second, 1 second, less than 1 second thermal decay relaxation constant in chloroform. And so this is not new in the literature. This kind of thing has been studied before by Asano and coworkers, and this is indicative of a change in the isomerization mechanism from a inversion to a rotation mechanism. The rotation mechanism is sort of stabilized by more polar solutions, and it's faster. So then the idea was to try to take these solution results and make some sort of congruence between the molecule behavior and the solution and what's going on with the monolayer. So if you just sort of scale the absorption spectra in solution to the number of molecules that you would expect on a surface, you can make these kind of plots. Obviously, it's not a very good interpretation of the monolayer. You can see that the trans-absorbent spectra of the monolayer in solution, that red curve, is significantly narrower in energy than the monolayer curve. In addition, if you use similar illumination conditions to what you would expect, would create a cis concentration in solution of, say, 65%. If you use similar illumination conditions on the monolayer, you get less cis. So, this isn't the best way to look at it, I guess is the point. You need a more complicated interpretation of what's going on with the molecules on the surface. In addition, the thermal decay constant for molecules in the monolayer is different than it is in solution. It's longer. So I guess then the point was to construct a better model for how the monolayer molecules, DMR molecules, are arranged and how that produces the optical signals that you're measuring. And this relation, this Clausius-Mosadi-Tech relation, tries to do that. And so this is relating the macroscopic optical quantities that you would measure for the monolayer to the microstructural arrangement of molecules in the monolayer. So it depends on things like the polarizability of the molecules, the effective thickness of the layer, the surface coverage of molecules in the monolayer, and the orientational information about the molecules in the monolayer. What's the orientational distribution function of those molecules? So this relates all that microstructural information to macroscopic optical constants of the layer. And this is just sort of a picture, sort of detailing the microscopic picture. And for this study, what I did was I assumed the Gaussian distribution for the molecules in the layer in the two angles of tilt that a molecule has from the normal to the surface, and then the azimuthal angle. Now, one of these optical constants for the layer you can directly access with surface absorption experiments, and that's good because it helps you to determine what the others are when you do more complicated experiments like ellipsometry, which depend on all of the optical constants of the layer. So the program, the experimental program, was to sort of study these layers with that sort of model in mind, and to then, as an experimental variable, try to vary the surface coverage of the DMR monolayers. So you start with a fully covered layer, essentially, or what's most likely a fully covered layer. And then you introduce into the surface attachment chemistry that puts this DMR molecule on a monolayer, or on a glass slide. You introduce a spacer molecule. So it has the same attachment chemistry, but it's, optically transparent and small enough that it shouldn't interfere with the DMR molecule. And if you do that, you can create these different samples that have sub-monolayer coverages of the dye. And that's what these absorption spectra show, is that we were successfully creating these kind of samples, and that the absorption maximum of these different monolayer samples, or sub-monolayer samples, I guess, technically, sort of varies as you would expect with the DMR, the concentration of DMR that was in the solution that created that particular sample. If you use that model that was from a couple of slides ago, you can sort of calculate the absorption spectra and other things about the DMR monolayer and compare that to your experimental results on these different coverage layers. And it turns out the model works really well for dilute sub-monolayer samples. So this is like the 1 to 4 dilution, where you have basically 20% of the amount of DMR that you have in a fully covered layer. And it works pretty well. Gets worse for dense films. So the other optical characterization that was used to study these layers is ellipsometry, and both reflection and transmission ellipsometry. And this just sort of shows you that you have to take into account these multiple interferences in your monolayer, which would be the slightly colored layer there. And if you do it all correctly, you can sort of, your signal depends on things like that you want to know, things you have control over, like the angle of incidence that your ellipsometry beam has with the sample, and then also things that you want to know, like the optical constants of that layer. So this is the kind of results you get from doing an ellipsometry experiment on these DMR layers, and you can see that, so what this is showing is the sort of blue and red points that are overlaid on top of each other are ellipsometry data for wavelength that's outside of the absorption band of the monolayer. So the monolayer is transparent at that wavelength, and so you get no change. But if you look at, the layer in the absorption band of the monolayer, then you can see that when you use an actinic source to, say, align the layer or to cause photoisomerization in the layer, you get a change in your signal. So that's what this is showing you. And then, and both, by the way, ellipsometric variables that you can measure. And so if you take this ellipsometry data and you take the absorbance data and combine it all, you can make tables of what that data means in terms of the optical constant variation of the monolayer. And so that's these red and blue lines on this. So this is a family of possible meanings that the ellipsometry and absorbance data provide for the optical constant. So that, we have the change in the imaginary optical constant, the imaginary part of the optical constants, versus the birefrequency of the layer. And so this is sort of showing you how those differences can vary with respect to each other for this monolayer. Now, superimposed on that, we have a curve that comes from this sort of microstructural Gaussian interpretation of what's going on with the monolayer orientation. And as you can see, all of this family of curves fall somewhere in the middle. And so the range of orientations available to this monolayer aren't that great according to the experimental data. So there's only a range of about 20 degrees. It's not like the thing stands all the way up or lays all the way flat. It's kind of somewhere in the middle, sort of centered around 50 degrees. And then the other thing the red and blue show is that, now this is for a dilute layer, remember. But the other thing that the red and blue curves are showing, what's different about the red and blue curves rather, is that the red curve is for the monolayer in the dark, and the blue curve is for the monolayer that's been illuminated. And so this microstructural analysis has been performed in both of those cases. And what you see is that the illuminated layer, the optical constants, don't fall very far away from the non-illuminated layer on this Gaussian prediction of layer orientation. So that means that the molecule really doesn't change in tilt very much when you illuminate it, maybe a couple of degrees. So that's an important result. Once again, this model does break down for denser coverages, so something else might be going on there. And then in terms of planar orientation, you can investigate the dichroism of the layer as a function of surface coverage. So now we're back into surface coverage and all these different surface coverages that we were able to create. And you look at the dichroism versus surface coverage, and you see that it goes. So this is the dichroism induced by illuminating the layer with polarized light. And so you can see that it goes up when you have greater surface coverages, quite a bit. The order parameter, however, for the surface sort of normalizes that effect out for the number of molecules that are present on the surface, and that's pretty flat. So that experimental result is kind of what you would expect. Now, this type of normal incidence, this comes from a normal incidence transmission ellipsometry experiment. That analysis was originally developed by young Louis, who was working up here at Dahdah. So this is sort of speaking to the planar orientation of the layers as a function of surface coverage. And then the other thing that comes from this planar study is the sort of times it takes for things to happen on the surface as a function of surface coverage. So on the top graph we have the times it takes to establish alignment in the monolayer surface as a function of surface coverage, and then the bottom part is the thermal decay of that alignment as a function of surface coverage. And you can see the thermal decay changes quite a bit. The time it takes to establish alignment in the layer doesn't change so much. It does change as a function of surface coverage, but not a lot. And then another thing you can take away from that top graph is that the time it takes to align the surface from a random orientation is faster. It happens faster than it does to say take an aligned layer and rotate the alignment. Just fully expected. So the conclusions are, I guess the DMR monolayering has atrophy and dynamics to kind of break down the nanostructure, and the model does provide some insight into that. The transabsorbance and thermal relaxation results suggest that there might be some kind of mean molecular field in the dense films, and that sort of is dissipated for the more blue films. The alignment layer switching speed is slowly varying functional surface coverage, so it's not the way to optimize LC cell switching. Inversion pathway is most likely the mechanism for the thin films. That's the relaxation mechanism. It's probably inversion. You don't get those 1 second times for thermal relaxation or anything like it in monomer. So that's it. And thanks for your attention.
Thank you. Is there discussion in this paper?
What's inversion?
Inversion is about the nitrogen-nitrogen double bond, and I think it just the bond inverts this way, so you can get that the cis shape without actual rotation about that bond. It's not like the orbitals are rotating about that bond. It's just
Before you ask your question, there's a lot of feedback. I don't know whether you can all hear us here. I think it'll go away if you. Is that any better? Try it out for. I mean, you ain't going to have that. I think she was standing further back. That's very unpleasant.
I was just interested in solvent effects. Early on you presented what you said was the absorption spectrum, the trans and absorption spectrum of cis, but that depends on the solvent.
Yeah.
Or are there strong solvent chromatic effects?
Not terribly. I'd say the variation from the solvent range that was discussed here, the variation in the peak absorption, just sort of moves the absorption spectrum to the left or right. And the variation is probably in the peak locations, somewhere around 10, 15 nanometers.
Right, and then these thin film experiments, have you repeated those experiments for both polar and non-polar solvents in contact with the monolayer?
No.
These are all done with non-polar and non-polar solvents then?
Well, these are all done in air. So.
Oh, air?
Yeah. Well, nitrogen. But yeah.
Nitrogen, okay, so there's no...
Yeah.
So the last, let's look at the last, yeah.
Okay.
So, for the dense monolayers, then it's 2 photons per reorientation.
Yeah.
2 absorbed photons per reorientation.
Yeah.
Okay, then, so then that number stays the same as you dilute.
So
We go to the next slide.
these are all at the same dosages. So yeah, all these curves, if you're going to plot versus, I mean, the illumination conditions are the same for all of these.
Yeah.
Yeah.
So that means that number stays the same, right?
Let's see. So
I guess.
yeah. Basically, yeah. Because the surface order curve. Yeah, sure.
Okay, so but then the time goes way up. The relaxation time goes way up.
Right. That's right.
So somehow that suggests that the relaxation is somehow collective, but the writing is somehow like individual molecules. Is that?
Yeah, it seems to
inference?
suggest that, yeah, whatever is going on, like say, if you pretended that there's a mean molecular field that controls the relaxation on the surface, whatever, the writing intensity or the effect of writing light, is strong enough that
doesn't
Pay attention.
pay attention to that.
Yeah, right.
Very interesting. I should just say that one feature of these, just a common one feature of these monolayers is that if you, which may not be, which may not be apparent from what's been presented, is that if you write the monolayer and then turn the light off, the birefringence goes away. Right?
Yeah, that's right.
But if you make a twisted pneumatic cell and say reorient it into the twisted state, and turn the light off, then it just stays there. So the collective coupling of the molecules that is affected by the liquid crystal contact is enough to sort of hold the orientation.
Right.
Other discussions of this contribution? Okay, if not, then we'll close this session, and the next session will begin after lunch, which is by yourselves or in groups, as you may see fit, and reconvene at 1.